We have always seen aloe vera as a medicinal plant, and it's also a plant which is less focused on research for research purposes. Assalamu alaikum warahmatullahi wabarakatuh. I am Firaz from Sana Model School, present here to deliver an amazing research on aloe vera as a biocatalyst. So, firstly, what is a catalyst? A catalyst is something which speeds up a reaction without even affecting the reaction itself. And there are two main types of catalysts. One, synthetic catalyst, and two, biological catalyst. Synthetic catalysts are widely used for industrial purposes, but they have lots of disadvantages. One, they cause pollution and they also lead to resource deprivation. It requires high temperature and high pH, and it's also costly. So industries seek eco-friendly alternatives to these synthetic catalysts, which are biological catalysts. So in order for something to qualify as a biological catalyst, we should confirm three factors. 1. It should be from a living organism. 2. It should contain enzymes. And 3. It should work at normal temperature. So firstly, I took aloe vera as the plant, and as we all know, it is from a living organism. So the first factor is confirmed. For the factor 2, we should confirm the presence of enzymes such as catalase and amylase. So the hypothesis of this project is, if catalase is present, then it will be able to break down hydrogen peroxide into water and oxygen bubbles. And if amylase is present, then it will be able to break down starch, leading to the fading of blue-black color when iodine is added to it. So for the catalase test, we need pipettes, test tubes, aloe extract and hydrogen peroxide. I took 3 test tubes with 5 ml of hydrogen peroxide. In test tube 1, I added fresh aloe extract. In test tube 2, I added heated aloe extract to see whether it can act in various temperatures. And in test tube 3, I added no aloe extract as a negative control and record the foam height. And for the amylase test, we require iodine, starch solution, aloe extract, pipettes and test tubes. And in each test tube, we should add 5 ml of starch solution. In test tube 1, I added fresh aloe extract. In test tube 2, heated aloe extract, and in test tube 3, no aloe extract as a negative control. And by adding iodine to it, we should record the color change. So what are the results? So in catalyst test, we are able to observe significant bubble formation in test tube 1, whereas in test tube 2 and 3, there is no bubble formation, confirming the presence of catalase. In test tube 2, we used heated aloe extract. So by heating it, the enzymes get denatured. And in test tube 3, as we used negative control, we are able to confirm that the breakdown of hydrogen peroxide was only due to aloe vera. And in amylase test, we can see that in test tube 1, the blue-black color fades into light blue, whereas in test tube 2 and test tube 3, there is no fading of blue-black color. Hence, we can prove the presence of the enzyme amylase. And as you can see, these are the tables. This is the table for catalase test, and this is the table for amylase test. We have proved the presence of enzymes. So where can we use it? So firstly, we can replace it with synthetic catalyst, which can reduce pollution, reduce resource deprivation, and it is also an eco-friendly alternative. Secondly, it can be used for wastewater treatment by breaking down hydrogen peroxide effluents from industries. And 3, we can use it in textile desicing. And 4, it can be used in various educational purposes. We can also learn about shell life. I got a question. Does aloe vera has antimicrobial effect? I performed the disk diffusion method to prove this question. So first, I took a bacteria sample and added it to agar petri dishes and incubated them under 37 degrees Celsius overnight for its growth. Meanwhile, I added antibiotic discs to aloe vera extract and placed the discs in the bacterial colony and incubated them. After incubation, we can clearly see a zone of inhibition, showing that the aloe vera was able to kill the bacteria. By this, we can confirm the antimicrobial effect of aloe vera. So where can we use this? So we can By this research, we can increase the shelf life of various food products, cosmetics, and daily use products. We can also reduce the dependency on synthetic antimicrobials such as preservatives. These are harmful to our health. So, for future enhancement, we all should try to make the effort by bringing it to use in large scale industries. So, in a nutshell, aloe vera was able to confirm all these three factors. It was from a living organism. We proved the presence of enzyme catalase and amylase. And three, the change in temperature led to the denaturing of enzymes. Hence, they can only work in a normal temperature. So, using these findings, we can confirm aloe vera as a biocatalyst. Jazakallah khair. Assalamu alaikum wa rahmatullahi wa barakatuh.